Okay, well, hello everyone. Thank you for attending this webinar with Crispin Given. My name is Pat Bono, president of New York Bee Wellness, which is an independent, educational, grassroots, charitable 501c3 non-membership organization. Its mission is to educate small-scale, sideliner, and beginning beekeepers. We do have a YouTube site with past in-person and online presentations. New York Bee Wellness also sends out newsletters several times a year, and we conduct statewide surveys twice a year for non-migratory beekeepers in New York. Welcome, Crispin. Glad to have you present and doing this. Thank you.
Yeah, thanks for inviting me, Pat. And I look forward to talking to the New York Bee Wellness Group. Sorry I can't be with everybody in person. I'm coming to you live from our molecular bee lab this evening. So I've been engaged in honeybee breeding now for about 18 years at Purdue. And I'm going to give an overview of that process that we've done in selecting for this trait. And I'll also cover briefly some other behaviors that are of interest that kind of lead up to this. Okay. All right, I got this meeting. So breeding for resistance, it's a process. So the first thing you need to do is you want to decide on a trait or a phenotype. And you want to develop an assay, then measure foundation population, and then select from that. And then use controlled mating using a design, and then you want to evaluate, document, and verify. So breeding versus queen rearing, what's the difference? Sometimes people confuse these two. Basically, with a honeybee breeding program or some type of paradigm, you need to have a process of traits or characters that you want to select for. So it could be something as simple as the color of the honeybee, or disease resistance, like resistance to chocolate. Whereas queen rearing, which most people do here in the US and abroad, is you simply just want to produce a large numbers of healthy, productive queens that beekeepers can use in their colonies. So healthy, productive queens is one of the most important factors to have in a honeybee colony if you want to have a successful breeding program. So just having a great queen makes a big difference. Many years ago, Dr. Rob Page mentioned his mentor, Harry Laidlaw Jr., who's considered the father of honeybee genetics. He was saying that just having a great queen is probably 80% of the process. And then the other 20%, maybe we can influence that through breeding, through selecting for certain traits. So the fundamental principle is that the offspring need to resemble the parents. So you want to have some consistency through time. So if you start with a population today, and you can expect decent results within 3 to 5 years, depending on the traits that you're selecting for. So you want to see consistency. So for example, the progeny, the color, the temperament on the comb, etc. So public enemy number one, we all know, is the Varroa destructor. And I won't talk much about that because I'm sure most of you are aware of it, but it is still the biggest problem. So where did Varroa come from? So they came from Asian honeybees, Apis ceranae. So these bees have dealt with Varroa for many years, and they have developed mechanisms to resist the mites better. So they're good groomers, and the mites are only able to successfully reproduce in the drone brood. And the drone brood of this bees is interesting. It's dome shaped like our European bees, but there's a little hole on the top, which is interesting. So they're able to have a symbiotic relation. They're happy, they can live happily together. Whereas the bees we're looking at today are Europeans. We need to select to get to that point, but it's going to take time. So decline in US beehives. I mean, when I was a kid in the 70s, it was easy keeping bees. The biggest problem was just trying to find new boxes to put your swarms in. I mean, probably, 6%, 12% was pretty much the highest we experienced back then. But through time, once tracheomites came in and Varroa, decline in populations grew. And then, of course, the so-called CCD 2007. I just put this up here to show how alarming this is. I mean, we are experiencing levels of mortality that exceed 40%. And this has been going on for the last decade. So this is a problem. And as I mentioned, Varroa is the main factor, but there's other factors. Of course, there's, pesticides, loss of habitat, modernity, we have all these new subdivisions popping up, and farmers are plowing their fields as close to the roads now. We don't have our fence rows anymore and things like that. So there's a lot of factors that are attributed to this. Surveys from the last decade, colony losses, of all the factors that could be measured, the presence of Varroa mites is the most correlated with winter mortality. And I might add, it's not only the mites, it's what we don't see that's causing a lot of mortality. It's the viruses associated with the mites, because the mite is like a dirty hypodermic needle, and it vectors these viruses into the hemolymph or the blood of the bees as it's feeding on the fat bodies. Many behaviors are influenced by genes. Here, a few of them here: stinging, pollen, foraging, undertaking, hygienic behavior, brood rearing traits, swarming tendencies, propolis collection, honey, and even learning. So here's some behavioral defenses. Put up a few here, because this is what I'm interested in, is looking at ways to combat the Varroa mite through behaviors. So we're all familiar, I think, or most people are familiar with general hygiene. And we also have the Varroa sensitive hygiene, and there's grooming behavior. And here at Purdue University, we've been looking at grooming and mite biting, but primarily the mite biting behavior. And there's undertaking. So hygienic behavior, sometimes this gets confused with VSH, but this is just general hygiene. So the idea is you want to test your best queens or your favorite queens if you can. And what we do is we test our instrumentally inseminated queens with this. We want to check their progeny. So what we do is we just use some soup cans. You can take both ends off, and about 250 mils of liquid nitrogen. And this is the freeze-killed brood assay. So you put these cylinders in here, and with plastic foundation, it's much easier. So you just twist it into the midrib, and once you twist it like maybe halfway, you stop. If you twist it too much, it can leak out and skew your results. And then about 24 hours later, you want to see something like this in your colony. Now you can also cut out a piece of comb and place it in your freezer overnight and reattach it into the surface of the comb. And that works well too, because not everybody can get liquid nitrogen. So hygienic behavioral is a heritable trait. Rothenbuehler found this years ago. So this is something that can be selected for, and it's something that's relatively easy to keep in your population once you have it. Another behavioral mechanism is SMR. How many remember? I'm sure some of you remember the old SMR, the suppression of mite reproduction. And then they changed it to the VSH, Varroa sensitive hygienic behavior. So this is the ability of the bees to detect anomalies in a brood, diseased or infected pupae, and remove them. So this trait is what we would call an additive trait. So there's several genes working in concert. For example, you might have one bee that comes by and recaps, and another bee comes by and inspects the cell and determines that the pupae or even the larvae is not infected, and then they'll recap it. So you have some bees that recap. So this picture on the far left here is this geographic look. You see some of the cells that are raised. Those are cells that they determine were okay. So it's interesting that they recap. So John Harbaugh here and Jeff Harris, they're the ones that were the genesis of this and selected out this trait, the VSH. And how they did it in the beginning, I'll just briefly mention, Jeff was telling me they made these mega packages. So they had like 100 pounds of bees that they did. I think they made several of these. So then they aliquoted out bees and from many different sources, and then they put them in these packages, and then they made colonies and started selecting from those. So it was a lot of work in the beginning, very tedious. Another example is response to pollen hoarding. So Dr. Rob Page did this for over 30 years, and he found, that you can select out, for high, they call this bidirectional breeding. So he'd have a high low and a high strain and a low strain. So he would look at those, and this was more so for scientific curiosity, but it was interesting through the years, all the results he got from that. So this is something that can be selected for too. So colonies that have too much of a trait, like this is an example of a frame that has the high pollen hoarding trait. Or it's kind of, you can think of it as a syndrome. So what's going on here is you have a colony that's in a single deep, and there's just very small amounts of brood, and there's large amounts of different pollens in there. So these colonies aren't productive at all. Now, grooming behavior, we're going into looking at the mechanisms behind this a little bit, and Ernesto Guzman and Miguel Archila-Nova, that they found that grooming behavior is important. So Miguel and Ernesto's study in Mexico, they found that grooming behavior was important, and those with the lowest infestation overall had low adult infestation. They also had the most chewed mites on the sticky boards. So mites chewed in the hives, they had Russians unselected, and they had a high-low generation 1 and high-low generation 2. Then they used Africanized bees, because African bees are also good groomers. Like Russians, they're already decent groomers without selection, as opposed to, say, Italians. So in the second generation, you can see that the portion of chewed mites was higher. And when they looked at the mites in the brood, they were relatively the same. But if you look at the mites on the adults, the low group had fewer mites. So there was a response to this selection for this trait. This is our 24th year of selection here at Purdue. So in 1997, we just started with VSH bees. We also got Russians, and we had stock from a wide geographic range, different beekeepers, so-called survivors. Bees that they thought did well in their area, and most of these bees were acquired from the northern part of the US. We also measured the mite drop and the strength multiple times in the year, the honey yield. And we just tested best sources for VSH and hygienic behavior. So the temperament was a factor that we looked at because nobody wants mean bees to work with. And then general disease resistance. And then in 2007, was in a lab looking, and I noticed some of the mites were chewed, and so Greg and I started selecting on the mite biting trait. So we started selecting for grooming behavior, and then we also select for chewed or damaged mites, because we noticed some of the mites that we were looking at under the microscope showed damage or missing legs or bitten sections of the idiosoma. And of course, strong overwintering ability, general disease resistance, and comb temperament were things that we looked at also. So we measured the Varroa populations with sticky boards. That's how we've been doing it. So we put sticky boards in for 2 days, and then we bring them back to the lab, and then we assess the number of mites on each board, and then we look at the proportion of chewed mites on each board. And we also do alcohol washes now. We've been performing those since 2019 on all the colonies. In the past, we just did a few colonies. In the past, we extracted survivors from a house. Of course, any bee that makes it through the winter, that's a survivor, right? But we did this, we did cutouts, and we took bees from beekeepers' hives that weren't treated, they claimed, and survived the winters. So collecting and counting chewed mites is tedious and time consuming. Unfortunately, I have an undergrad that help us do this every year now. So we basically bring all these mite boards into the lab, and then we have to evaluate the grooming behavior. So what we do is we remove the mites, we place them on glass slides on their backs so we can see the presence of chewing much easier. Because if you have them on the ventral side down, it's much more difficult to see that. So here's some chewed Indiana mites. I like to see it when the shell, or the shield of the mite is chewed, as you can see here, but most often we see legs missing, and so that's pretty classic mite biting trait there. Here's Greg Hunt. He's looking at the mites here. And here's what they look like after they've been collected and put on the slide. So as you can see, it's a lot of work and a lot of time. So each one of these glass slides represents a colony. So we put 100 mites on there. So if a colony has, say, 300 mites, we arbitrarily only collect 100 of them from that colony and put on the glass slide. If it has 100 mites only, then of course all the mites are placed on the glass slides. And here's another picture. It's kind of an abstract image. Here's a really nice photo here we got of showing the appendages missing. You can see here very often the pedipalps are missing and legs. Here in the center, you can see the center portion of the shell's missing. So I like to see that when they bite the shell. It seems like it's more aggressive behavior in response to trait. So some challenges with the chewed mite assay is you need to have enough mites. So often in the springtime, we don't really see that many mites. In some cases, there's none, because most of the mites, once the colony goes back into reproductive mode, 80% or more of the mites are going to be under the cappings. And so that's a problem. And poor repeatability. Sometimes there's unknown factors that cause an uptick or a reduced amount of chewed mites in the colony. Then there's correlative traits or correlative characters that can also affect the trait. And of course, history of the colony is a big factor. So if the colony was split, or if you made nukes from the colony, that's going to put a break in your brood. And that's also going to put a break in the reproduction of the Varroa mites, or if the colony swarmed, or if it superseded its queen. So these things need to be recorded in your notes. That way we know exactly what happened. You need to have enough colonies. So if you're seriously thinking about starting a bee breeding program, it's good to have at least 100 colonies. So this is one of the challenges for a lot of people. They don't have the resources or the time to manage that many colonies effectively. You need to try and control the matings. So try to flood your area with your type of drones that you have. And that really can help out a lot. And mark the queens, so that way you know if there's an unmarked queen in there, for example, you know that it's the daughter. I've had it many times where I'll have daughter of the daughter. It'll be two replacements in 1 year. So that happens quite a bit. So keeping good records is important. We use hide-to-hide chewers, and then we cross those. So the best ones, the hide-to-hide, the high miters, mite biters against the high mite biters. So we cross those two together. So we use controlled mating with instrumental insemination. So this is paramount. You need to, if you really want to control, unless you're fortunate to have a isolated area or an island, which we aren't. So we have to perform instrumental insemination. But this can also speed up the trait selection too, if you're doing this. So daughter queens and drones are generated from those queens that are produced. So how do you choose the parents? You need to evaluate and keep good records. How do you control the matings? You need to perform instrumental insemination and have a good drone source for flooding your mating yards. This can really be, this can have a big influence if you have a large number of your type of drones. And you can do this by putting drone comb into your colonies, your favorite colonies, and that increases the chances of those queens mating with your type of drones. You need a trait you can evaluate, something that's beneficial and something that's heritable. So the take-home message with this talk is these traits are all heritable that I'm talking about. So it's possible you can select for these from your bees that you have in your backyard. So start with bees that have good traits for your area. So here's what we do every year. I paint mark. I like to paint mark the drone sources. That way I know what lineage they're from and the age of the drones. And typically this catch them on the wing as they're returning to the colony, and then bring them into the lab to pull the semen from them for inseminations. So here's a look at the paint mark drones. So I could mark, a couple thousand drones within about 2 hours. I typically do it in the morning. They're not as runny in the morning as they are in the afternoon. So they kind of have a photo period. Even when they're young, they're more excitable in the afternoon. So in the morning, they're sluggish, and drones are slow to mature. So it's easy to identify the ones that have emerged within, the last two days. So this is just a picture of our nucleus colonies that we use. So we use 3 framers and 5 framers, standard length frames. And that way everything's standardized. So selecting for mite biting is considered, you can say it's a hierarchical selection. So we measure proportion of June mites. Did we eliminate those that show disease or did not control the mite population growth? And we also eliminate those that do not pass a freeze kill hygienic test. And then we select our breeders based on that. So for me, bee breeding is kind of a road with no ending. You can't sit back and enjoy the fruits of all your hard work because certain traits can be lost quicker than others. And it's kind of like walking a tightrope. Also, you have to keep your eye on the rope and keep balancing yourself. And it's easy to fall off and lose traits if you don't. So constant selection is the key to any honeybee breeding program. So something else we do is we evaluate our II queens. So over here, sometimes we put them in a cage. We can evaluate, we know what progeny are theirs. And we can also see, how well the queen is performing herself. But in some cases, you can have, for whatever reason, an instrumental enzymatic, the progeny don't always perform like you want. So you can have deleterious effects or deleterious alleles for whatever reason. But most often you'll see an uptick once you get your program started. Another interesting way of testing for VSH behavior in your colonies is you can take drone foundation or drone comb, put the drone comb into a colony, an unselected hive like an Italian hive that grows a lot of mites, or a colony that has a lot of mites. Put that in there, and then once the pupae are capped over, you can place that into one of your breeder queens from sources that you think are good. So this is what we did, and about 48 hours later, this is what we saw. So you can see there's quite a few pupae removed, and you look at these white cappings here, you can see what's happened here. I call this crowning. So they've removed the wax and they're at the cocoon and they're in the process of removing those. So had this been left in another day, those would have been removed too. So this is a really good barometer for measuring your population too. Through this picture back in here, how many people have seen this? This is a classic VSH frame. And this is kind of VSH. Again, this is, in some cases, you can have too much of one trait. Here, this is kind of VSH gone wild. So we got VSH in our bees, and sometimes, occasionally, we will come across a frame like this where they're just uncapping and recapping, and they're just really going to town on it. And a lot of cases, these colonies aren't as productive. Another challenge or problem with bee breeding is you have loss of introduced genes. So here I just kind of put through time, you can lose traits. I mean, some traits, yes, you can lose quicker than others, but within 10 generations, you can lose a lot of selected traits that you're looking for. So mite biting is a heritable trait. So in 2007, when we started selecting on mite biting, we were just around 5%, and then around 2015, we were upwards of 50%. Here, this just shows that colonies that chew more mites also have less mites on the sticky boards. A few years ago, there was a paper published, and bees, they bite for a reason. We used to think that when a bee bites and she releases this compound, it's called 2-heptanone, and we thought that was associated with defensive behavior, as being an alarm pheromone. But it turns out that it's a paralyzing agent, and so bees bite for a reason. So when we came across this, we were excited because it kind of ties in a little bit with what we're doing. So bees bite for a reason. A recent publication, it's open access. If anybody wants to Google this, you can read the paper. We found grooming behavior gene expression of the Indiana mite biter honeybee stock. So this, again, you can see this. So in that paper, we found, basically Indiana mite biters, they had fewer mites than the Italians. So the proportion of chewed mites was also correlative. So you could see that the gene expression, for example, in direction 1 is more, if the ones what were grooming more and biting more, there's a synaptic response in the brain of the honeybee. So it was higher in those bees that were grooming more. And there was also a correlation to the percent of chewed mites also with the grooming, which is nice. So here I put a video. I hope this works for you guys. So we have 2 bees in here from 2 different instrumental and simiate queens. And we placed a Varroa mite on their backs. And we wanted to see how they responded in the lab. So we call these surrogate cage assays, or you bring them into the lab and you want to look at them and verify a trait. So here you can see the lime colored bee is responding more than the green. You would think with something the size of your fist, if you look at it to the proportion of our body, it'd be like having something that's big as your fist on you. That bee would respond. So it's interesting. Some bees just don't respond as much. And we're not really sure why. Through the years, we've tried many other assays trying to look at other ways to measure mite biting and grooming behavior. One that really seemed promising a few years ago was we were placing flour on the bees. And the first 2 trials, there was a correlation to the field, to the colonies. But then on the 3rd trial, it was all over the place. So I guess that's why they call it research. And then we've done cage assays too, where we've placed individual mites in cages and see how many they groomed off. And we had improvised cup cages and things. So it's difficult trying to develop an assay that beekeepers can use. One study here that kind of similar to a previous study, Indiana mite biter versus Italian stock. So we looked at the mite population growth, the proportion of mutilated mites, the severity of mite mutilation, over winter survival, and the amount of neurexin expression. So 18 colonies were split in 1 apiary and then sampled July 10th, only 25 days since the split, and October 11th. So we assessed for mite drop proportion of mites and the amount of viruses that were in their blood. So grooming behavior and mite levels. So here you can see in the Italians, the percent of mite drop was higher and the percent of chew was significantly higher in the Purdue's versus the Italians. And in October, the same thing, we noticed there was more chewing going on in the selected Purdue stock versus the Italians. Some viruses that we detected was DWV, black and queen cell, like cyanide 1 and 2 virus was also probably. But DWV is by far probably the most devastating virus still. So DWV correlated with the mite levels. So survival as of February, the Purdue's were at 77%. The Italians were at 22%. And the Indiana mite biter, if you look at the Varroa population, again, it was much less than the Italians. If you look at the proportion of mutilated mites, and again, it's much less than the Italians. And the Indiana mite binders over here, if you look at the proportion of mutilated mites, it's less. So bees are fighting back, and we're trying to just help bees through these behavioral traits to improve the health of the colony. Now back kind of to the fundamentals. I mean, we're also at Purdue, we're teaching beekeepers how to produce their own queens. So it's important, you know, yes, we're doing the bee breeding and all this, but it's also important that beekeepers just have the fundamentals down, you know, the practical applications of beekeeping, just taking care of your hives, producing your own queens. That's very important, because today's world, we kind of forget about, you know, just the fundamentals. So here's a group, we have about 40 beekeepers every year that come to the university. We teach them this queen rearing class. And we also teach the same class at HAS, the Heartland Agricultural Society. And this year they've requested that I accept 40 students, so I have 3 other instructors helping me. So that's already booked up for HAS, I believe. So that's a lot of fun too, working there. This year it'll be in Sens, if you just Google HAS. So hope to see somebody be there, but it's a lot of fun. We just teach beekeepers how to produce their own queens, and that's kind of a big factor that helps out a lot. And we also teach instrumental insemination each year at Purdue. And this is more for beekeepers that are, you know, they want to step up their game and they're serious about breeding and they've learned queen rearing and all that. And so this is a lot of fun also teaching them this. We also have collaborative efforts with beekeepers, or we'd like to call them micro breeders. And one of them is the Heartland Honeybee Breeders Cooperative. So we have like 7 and 9 states that come to the university each year, and we disseminate information and germplasm or queens from our lab that they use, and then they take it back to their states and they produce daughter queens from those. So it's really unique situation where all beekeepers work together like this, and bee breeders. Another one in our own state, we have the Indiana Queen Breeders Association. And within our state, we have they have a website you can go to, and there's a lot of enthusiasm about queen rearing and bee breeding in our state. So this is last year's event. That's a lot of fun. Some future plans, we're going to do a collaborative effort in about 7 states, more stock comparisons. And Dr. Brock Harp, who joined our lab in 2019, he's interested in genotyping all of the mite biters. So someday maybe we can apply genomic selection in addition to our phenotypic selection. Because in the past, we've always done strictly phenotypic selection. So now we're going to incorporate genotype. So we'll do genome selection also. So that's exciting. Continue teaching queen rearing and expanding infrastructure and queen production selection. And then we have a PhD student, Garrett Slater, who's working on drone sperm viability and mortality associated, what genetic components are associated with those factors versus environmental ones that we're aware of that cause sterility in drones. So you can check out our website. It's the Beehive, we call it. You can kind of see what we're doing here. It's real exciting what Dr. Harper's doing, and it's going to be interesting to see what's happening in the future as far as looking at genes or finding new markers associated with these traits. So I'd like to thank everybody for listening, and just a few people here I added through the years have helped us a lot with this. So thank you very much.